Okay, so cocoon versus skin, water saving science of frogs. So imagine this. You're suntanning on the beach and you begin to sweat. After sweating for a little bit, you begin to feel dehydrated. You grab a bottle of water and drink it to help combat your dehydration. This model is similar to what frogs do in dry climates to prevent excessive water loss. Frogs lose water through their skin, which can be a major issue in dry places. Some frogs, such as the Australian hyalid frog, can lose less water from their bodies by forming cocoons. These cocoons are made from shed skin and special body secretions. Researchers are interested in understanding whether fats, also called lipids, in the frog's skin or the cocoon help reduce water loss and how these fats work, especially when the frogs are inside their cocoons. So we have a biological question, and a biological question is a question that focuses on understanding living organisms and their functions. Our biological question is, do fats help frogs lose less water through their skin? If so, where are these fats located and how do they work in the frog's cocoons? We came up with some hypotheses. A hypothesis or a hypothesis is a testable statement that predicts the outcome of an experiment based on prior knowledge or observations. Some hypotheses are lipids help frogs lose less water through their skin. Lipids do not affect water loss in frogs. Lipids in the skin have less of an impact on water loss than lipids in the cocoons. Frogs inside cocoons have frogs inside cocoons lose less water than frogs outside cocoons, or frogs inside cocoons lose more water than frogs outside cocoons. And just to reiterate, lipids are the same thing as fat.
Frogs rely on their skin for both respiration and water balance, which makes skin water loss an important trait to study, especially in terms of climate change and dehydration. So, to measure this, scientists use open flow through respirometry, which is essentially where dry air is pumped into a chamber with the animal in it, and they analyze the after exits the chamber to determine how much water vapor has increased. This is telling us how much water that the frog is losing through their skin. And then if you want to separate this total body water loss from respiratory water loss, which is how much water they're losing by breathing, you use the same method but factor in the volume of air exhaled per minute. So, continuing on with methods, the scientists took subsamples from the skin and the cocoon and freeze dried them, and they did this so that they could measure and compare the weight before and after organic extraction to determine how much dry weight versus the lipid amount was. So, freeze drying it is basically removing all of the water and the liquid from the skin and the cocoon. And then scientists used thin layer chromatography, which separated the lipids into bands using solvent and a plate. And in the solvent, the lipids will move up. So the location on the chromatography plate will correlate to the type, as certain lipids travel slower than others. The best way to sort of visualize this is imagining if you spilled different drinks like juice, soda, and syrup on a paper towel, the liquid will spread upwards, but not all of the drinks move the same way. Water will spread a little bit more quickly versus syrup will barely move because it's thicker. And thin layer chromatography works in a similar way because you can see the separate types of lipid classes in different areas of the plate. And you can use this thin layer chromatography plate and scan it using photodensitometry, which is a scanner which will assess how thick each band is, which is somewhat similar to if you drew on a paper with different crayons with different colors, and some lines were thin and some lines were thick and dark, and then you shone a flashlight underneath it, and the flashlight will basically show you how thick the bands are, and it's a similar thing with the lipids. So this is table 4 from the paper, and this is an important paper or an important table from the paper because it shows a breakdown of the different fat types in the animal. So as you can see, there are 1, 2, 3, 4, 5, 6, 7, 8, 9 different types of lipids, and cerebrosides and ceramides are the most important for water loss, as we'll later talk about. These are types of fats that humans also have in their skin, which are important for water loss in humans as well. And a similar table for skin versus the cocoons showed a similar lipid breakdown, except for in frog skin, they don't have cerebrosides and ceramides. So it's this table is essentially comparing the different types of species to the fat type and amount. And then this next table is basically showing cutaneous water loss, so water loss through the skin, when the cocoons are kept intact. So that's when they still have their lipids versus the cocoon discs, which don't have lipids in them. And as you can see from the left to the right, the numbers are increasing, meaning that as you are removing these lipids in the cocoon is losing more water. And so these are the 5 main results from the paper. Number 1 states that water loss was significantly higher without a cocoon and bigger the more surface area a frog had. That figure is something we'll go on to explain our interpretation. But the larger the frog is, the more skin they have, and the more skin they have, the larger like surface area they have to lose more water. Number 2 is for every species measured, water loss increased when their cocoon had lipids extracted from it. So when you don't have fat to protect you as a layer from losing water, you lose it faster. Number 3 is the more total lipids an individual had in their epidermis, the lower the CWL. So the more of this protective layer that you have, the more effective it is at preventing water loss. Number 4 is the more cerebrosides and ceramides in the cocoon, the less water lost. So these are the two really key fats for water loss. So when the frogs are in their cocoons and they have these two fats protecting them, they're less likely to lose water. And number 5 is other lipids such as free fatty acids had a minimal impact on water loss as a whole. So these other lipid classes that we saw on the previous slides, although their total amount is important, their type is not nearly as important as the cerebrosides and ceramides.
Doing that open flow respirometry, the which again was the flow of air over a frog that was sitting inside of a tube and then measuring the amount of water vapor that came out of the other side, allowed them to measure how long a frog would last underground before losing 30% of its body mass by evaporation. So when they tested this, they found that the frogs with a cocoon lasted far longer without losing as much water, which shows that cocoons are responsible for retaining water and containing things important for retaining water that the skin isn't doing sufficiently. So there's something special about the cocoons that the skin isn't doing. Then scientists found that cocoons contain less fats per volume than the skin, but this cocoons contain types of fats that the skin didn't. As you can see on this graph, the cocoons again contain something different than the skin, which is allowing them to maintain that water. Together, these suggest that the fats contained in the cocoon and not in the skin are responsible for preventing that water loss because, again, like I said, the cocoon, frogs with a cocoon last longer than just their skin. And the cocoons contain something different than the skin, which is the ceramides and the cerebrosides that we talked about previously. So these studies suggest that it is the ceramides and the cerebrosides that are leading to that higher amount of retained water because they're present in the cocoon and not in the skin itself.
So, regarding our conclusion, the main takeaway from this study is basically that frogs inside cocoons lose less water compared to those outside of cocoons. It's important because water retention helps them survive in the really dry environments, like we previously mentioned. And like we stated in our results in our interpretation, the reason for this difference is the composition of the fat. So the different types in the frog skin versus cocoons is what really matters. And these can be attributed to the ceramides and cerebrosides that were just previously mentioned. Another fun thing about the composition that we see in cocoons that are doing the most work when it comes to water loss is that fats in other mammals and birds specifically have a really similar function. So, for future ideas of where this type of research can go, we could conduct comparison studies, which basically compare the types and the proportions of fats that are found in frogs and other species. Functional studies, so isolating specific types of fats and looking at them and testing how they can contribute to water retention across a bunch of different species. And this type of research can ultimately contribute to conservation research, which is really important if we're looking at climate change and how issues like that are affecting different species and the type of methods that they have to adopt to adapt to rising temperatures. This type of research could also be really significant for looking at evolution and trying to find common ancestors between different frogs or different species based off of the way that their skin or cocoons are kind of composed when it comes to these different kinds of fats. And this is our works cited.